Part 3 of the nervous system embryology, the final part. In the last video, or in this last video, we're going to discuss neural crest cells. Well, we might touch on some other things too, but we'll primarily focus on neural crest cells. So let's look back at our timeline. Notice that neural crest cell migration happens pretty early on. Oops, up here, right? On day 22, as we start to get initial fusion of the neural folds, those neural crest cells begin to detach, and they form clusters between those two ectodermal epithelial layers that we've discussed previously. You can see in this timeline that our PNS will start to form, right? We see spinal motor neurons, we see ventral roots, we see our autonomic nervous system structures, ganglia. Those structures begin to develop. So this all starts to happen at the end of week 4 when we develop dorsal root ganglia, those roots that form our spinal nerves, and then these peripheral parts of our autonomic nervous system. Keep in mind that a large chunk of our peripheral nervous system is formed from neural crest cells. Let's take another look at the neural tube as it's forming. In the images on the left, we see the primitive streak and primitive node in figure A above, and I'll just highlight those in green. Our primitive node, and then our primitive streak right here. In the top right image, figure B, we see that we've formed endoderm already. Right here, we've formed mesoderm, and we've formed overlying ectoderm. And we can also start to see the development of the neural plate and the elevations of the neural folds in this top image, right? We're starting to see neural folds forming right here. In the middle right image, image C, we see the neural folds with a well-defined neural groove in the midline. And we can see that the folds are starting to come together here. From these folds, we have cells that delaminate. They basically undergo an epithelial to mesenchymal transition, and we lose the cadherins that are binding them so tightly to neighboring cells. We end up with groups of cells that are now capable of migrating. Initially, they migrate just lateral to the neural tube, and then they'll migrate outwards to other parts of the embryo. It's a combination of both permissive and inhibitory signals in other parts of our body that determine where specific groups of neural crest cells will go. So it's important to understand that neural crest cells detach from neural folds along the entire length of the neural tube. Some move to specific places, others go someplace else. Intercellular signaling is incredibly important for these groups of cells to end up in the right place. Here's an example of the range of structures that develop from groups of neural crest cells. Much of our endocrine system contains cells that are derived from neural crest. For example, the cells within the adrenal medulla are derived from neural crest, these chromaffin cells. We also have cells within our skin, the melanocytes, that are neural crest derived. As I mentioned before, much of our peripheral nervous system is formed from neural crest. This includes structures in the somatic nervous system, like dorsal root ganglia, as well as structures in our autonomic nervous system, like the sympathetic chain ganglia and the prevertebral ganglia. There are even structures within our heart and gut tube that are composed of neural crest cells. Don't forget that many of the bones and cartilages of our head and neck also develop from these cells. Seeing this, you can probably understand how a problem with neural crest cell migration might include defects in the heart and gut tube in addition to craniofacial abnormalities. When we examine the length of the neural tube and look at the groups of neural crest cells detaching from neural folds in specific areas, we see that each group will be stimulated to migrate to particular locations. Neural crest cells in the cranial region will become some of the neurons within the cranial nerve nuclei, in addition to structures in the eye, like the iris and the ciliary body, structures in the skin and teeth, and our pharyngeal arch cartilages. Head neural crest also become parasympathetic ganglia associated with some nerves, like the one associated with cranial nerve 3, and we'll talk about these when we get to autonomic nervous system. We have vagal crest cells, neural crest cells, that follow the pathway of the vagus nerve, and they contribute to some structures innervated by vagus, such as the heart and parts of our enteric nervous system, which is the nervous system regulating our gut tube. Specifically, our vagal neural crest will become cells that are within the upper portion of our gut tube. The rest of our trunk neural crest will form peripheral nervous system structures like the dorsal root ganglia and the sympathetic ganglia and support cells for those. In the lumbosacral region, neural crest cells will contribute to formation of cells within the lower part of our gut tube. So again, we're forming our enteric nervous system. In class, we'll look at a few specific cases, maybe clinical cases, and we'll apply some of this knowledge. So I'd like you to be prepared to talk about which groups of neural crest cells develop into these specific structures.